Hello everyone, I'm back again with the next video of Chromosomal Theory of Inheritance, chapter Principle of Inheritance and Variation, class 12. NCRT is being followed along with few PPTs. The topic that we will be doing today is chromosomal theory of inheritance. Now the question here children arised was, why did Mendel remain unrecognized, or his work was not recognized for years, even though today we call him as the father of genetics? Why is that? So this question generally comes, and here in your NCRT they have given you few lines. You can underline and mark. You can see here the reasons. Firstly, they start from here. So this is your first point, why Mendel's work remained unrecognized. So firstly, why was he unrecognized? Because of the communication was not easy in those days, and his work could not be widely publicized. So this was the first point, communication was not easy. So you can make points why Mendel's work remained unrecognized. This was your first point. Secondly, they say in NCRT, his concepts of genes or factors are as stable and discrete units that controlled the expression of trait and of the pairs of alleles which did not blend with each other, was not accepted by the contemporaries as an explanation of apparently continuous variation seen in the nature. Now children, this is very important. You know what Mendel said? Mendel said that genes, or what he used to call as factors, we also call them as Mendelian factors. Okay, genes were known as Mendelian factors. So Mendel thought that, or he said that they are stable. They are stable discrete units. That means discrete unit means they are small units, smallest units, and they are stable, and these genes control the trait. These are the genes which will control the expression of the trait, any character. So genes control any character or trait, and he said that they are stable, which means that if there is a character, suppose we say that pea plant have round and yellow seeds, and according to Mendel, if they are stable, then for every generation we should get round and yellow seeds because the genes are stable. We should not get any variation, right? As he said that genes are stable and they are the discrete units which decide, these are the things which decide the character or the trait, then according to this we should not see any variation. But you saw that during dihybrid cross, even though the parents were round yellow and wrinkled green, still we got two such varieties which were not there in the parents. So if Mendel's point is true, if Mendel's concept was true, then how did we got this variation? So we are getting lot of variations in nature. We are seeing lot of variations in nature. So what about that? Mendel could not explain this question. So see, this is our point, that the concept of genes or factors as stable and discrete units could not explain the continuous variation. So people asked Mendel that tell us how variation occurs, because according to you genes are stable, so there should be no variations. Then how these variations have occurred? Mendel could not answer this question. So this was also a factor why he got unrecognized. Then children, you see here in your NCRT, thirdly, Mendel's approach of using mathematics to explain biological phenomenon was totally new. People were not reading biology by using mathematical calculations. It was a very new concept in biology. And so, now when there is change, it is very hard to accept the change. You have seen now in COVID-19, we teachers are trying to teach on this platform as I am doing. So when we started with this, there were many parents and many other teachers who said that, oh, this is not the right way to teach, but now everybody is doing and learning. So yes, the things were unacceptable. What was unacceptable? The use of mathematical calculations in biology. So that was also a reason. You see, his mathematical approach was new and unacceptable. Then finally, the last point here, children, see in your book, though Mendel's work suggested that factors or genes were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. Though he was talking about those factors, though he was talking about the genes, but he could not prove or provide its physical proof. He could not show any experiment, or he was not able to physically show anyone a particular portion or a particular factor or a particular gene so that he could prove the points that he was saying. So last point, he could not give physical proof for the existence of factors or genes. So these are the points which Mendel, which were the drawbacks, you can say, for Mendel, and hence he could not, he was not recognized, or he remained unrecognized in the field of genetics. Okay, now you see here in 1900, three scientists, children please underline, because in generally in boards we are getting such one marker questions as who discovered or who invented the various concepts or theories or organs or parts. So you should also underline the names of the scientists who did that. So three scientists were D. Veris, Correns, and von Teschmark. Okay, these are the three scientists who independently, they did not work all together. They did not work together. They were working independent of one another. So they independently rediscovered Mendel's result. That means whatever Mendel had discovered, all three scientists rediscovered whatever Mendel said on the inheritance of characters. Also by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division. Now because this was time of 1900 and Mendel, it were many years had gone, so new discoveries were coming on, advancements of technology was going on, so microscopy also came into existence, so they were able to see cell division. Now the scientists were able to see cell division. So this led to the discovery of structure in nucleus. They now could explain the structure of nucleus. They appeared to be double and divided just before cell division. So because they were able to observe cell division, they said that the nucleus in a cell, when a cell is just about to divide, it is having two nucleus because karyokinesis has occurred, and now the cytokinesis will occur. So these double set of structures which they observed, the double set of structures which they saw were chromosomes just before cell division. Okay, how did they observe it? They stained it, and hence they were easily visible because of the stain their scientists did. By 1902, the chromosomes movement during meiosis had been worked out. So see, after 2 years, in 1900, they could see chromosomes. Then after 2 years, they said that chromosomes could move during meiosis. Then children underline this also, Walter Sutton and Theodore Boveri noted that behavior of chromosomes was parallel to the behavior of genes. That means they were almost similar. The whatever behavior was observed by genes, chromosomes had the same behavior. So that means they were parallel to each other and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosomes during mitosis, during equational division, and during meiosis, during reductional division. The important things to remember are that chromosomes as well as genes occur in pair. Now, because I said that both of them are parallel to each other, why are they parallel to each other? Because they both of them occur in pairs. Chromosomes also occur in pairs, genes also occur in pairs. Okay, so the two alleles of a gene pair are located on homologous sites on the homologous chromosomes. So to explain this, I will be using a video so that you get through this explanation very clearly, and it will be a piece of cake for you when you go through that video. So this I will be discussing in the next video, which I'll be posting soon. Okay.